as we would hope to maximize profit in the fishery as well as maintain the population. So we're okay, we can no longer blame the fishers. Here we could. This is all overfishing. So when you exceed the threshold here, then we're overfishing it. We were constantly overfishing it. So that's why we thought that, okay, let's impose the management actions and we're going to do okay. And we did okay for a while, but now all of a sudden we have some other environmental disturbances. So let's go to big data. Okay, so now I'm going to show you the kinds of data we have, and I will say that we are just beginning to analyze this. So this is one of those things where if anyone is interested in trying to work with us on that as part of a project, thesis, whatever, I welcome you to do that. So I'm going to show you the kinds of data we have and where we're just beginning. So this is the initial sampling, 1,500 stations. We get the blue crab abundance at different stages, salinity, temperature, dissolved oxygen, bottom types. I'm going to show you some of the kinds of data that we have. This is one of the things. This is the F1 plus means females that are 1 year of age and older. That's an F1 plus. It's just a stock assessment term. And this was the winter of 2009 and 10, December of 2009 through March of 2010. And this is the crabs per toe that we were getting. And it ranges up to 50, from 0 to 50. And so basically we have this information then for females, we have it for mature males, we also have it for juveniles. The juveniles are primarily in the tributaries, as are the males, whereas the females have already migrated down here. They're overwintering before they hatch out the larvae. They spawn and hatch out the larvae the next spring and summer. So this is one type of data set, and there are different ways of analyzing it, but basically the kinds of information you can get is, are there changes with fishing pressure? Are there changes with different habitats? Are there changes by year as far as the number of mature males and females, say in Maryland versus Virginia and so on? I mean, it's really wide open. So let me show you some of the other kind of data. So this is one of the things that's been concerning us is climate change, global warming. And we have seen this in the Chesapeake. The Chesapeake is slowly warming, and at this point, we don't know what its effects are. So, but we are starting to see, this is just a very first simple look at the effects of temperature. And what this shows is over about a 10-year period, it shows the abundance of crabs as a function of the first day that the temperature exceeded 10 degrees centigrade. 10 degrees centigrade is the temperature at which, sort of the threshold at which crabs become active. And so when you're below 10 degrees centigrade, that's when we know we can catch them, we're comfortable with it. And so we want to see where they are relative to the day at which, actually the day at which the temperature dropped below 10 degrees centigrade in the wintertime. So some years it might stay warm, you know that in December you might get a day in the 70s and so on, for several. And other years it gets cold right away at Thanksgiving and it stays cold. And so one of the things that we've noted is that depending on the day, so that's the day of the year at which we hit 10 degrees centigrade. So the 377 just means that we hit it in January. We start in 1 year, and then 377th is the 12th of January. Okay? And so what you can see from this, just initially, just a visual analysis, that in those days when the temp was cold early, we got most of the crabs were still in the tributaries and so on. As it got warmer, then you start to see more of them further down. They probably had the opportunity to keep moving since it was still warm and they could be active. And so this is one of those sort of types of big data where we're just sort of seeing that, but we don't know what it means. We don't really know at this point what the importance, what the implications are of these kinds of patterns. Are they going to affect the way the fishery operates and therefore mortality? It also would affect the overwintering mortality. We can have a mortality in Maryland, for instance, of up to 30% of mature males in Maryland when it's a severe cold winter. Remember, the blue crab is tropical in evolutionary origin, so it likes warmer temperatures. And so we've got changes going on, and we don't really know at this point. And the changes are not just in time, they're in space. There's shifts in the way the population distributes itself. Okay, so this is some of the biological data. Some of the other data that we collect, this is a sediment map. So this is just of Virginia down. And so at every one of our sampling points, we do what's called a grain size analysis. We take a sample of the bottom, and then we analyze the data. We analyze the sample for the percent sand, percent silt, and so on, and that just shows you. So wherever it's green, it's low percent sand, which means it's primarily muddy. So these areas are primarily muds. And then it switches over to primarily sand in where it's red. And it turns out that there's several interesting things about that. The crabs, the winter dredge fishery, when it operated, causes much more damage and injury to and mortality in the crabs when it's over sand, because the teeth can't dig in as far and they end up slicing the crabs. And so that's waste. They also slice horseshoe crabs, which are really important for birds. Their eggs are really important for red knot, for instance. So that's bad. That's why I actually did that in red, whereas in the muds, the teeth dig down and get the crabs whole and so on. Anyway, we have all this sediment data, plus the crabs like to distribute themselves in some fashion according to the sediment regime for overwintering and so on. There are also some other things that go on in the bay. And again, we have this farther up, but how many of you have heard of the dead zone in the bay? Anybody? Okay, so every year, because of the high nutrients that we push into the system, then you have what are called algal blooms, microscopic plants. And when those die, they settle to the bottom. And then when they settle to the bottom, their degradation by bacteria uses up the oxygen, and that's what causes the dead zone, low oxygen. And that happens primarily here down to somewhere around here. So again, we've got layers just like this of dissolved oxygen. Of course, that affects not only crabs, but affects all the species they eat, worms, clams, and so on. So all that information is available basically on a bay-wide basis. And if you hear about any of the attempts to regulate nutrients, farms, and so on, that's why, because everything coming into the bay is basically used up by the plants, and then we have the dead zone. Okay, so that's an issue of habitat quality. Class ends at 2:50? Yeah. Okay. Some of the other information that we have, seagrass is a nursery. Those are little snails on seagrass, and that's eelgrass there. Eelgrass is probably affected in a major way by global warming, by the way, because it's a temperate species. We're near the southern limit of its range, and it may actually be extirpated down the road. This just shows the distribution of seagrass, but how close we've come to restoration goals. As you can see, this 0 to, I believe, 20%, 25%, and this is up to 50%. So most of the bay hasn't met even 50% of the goal, somewhere between 0 and 50%. So there's been a general reduction. Global warming is also affecting seagrass. So that kind of information is available to relate to crab data. So basically, this is sort of the end of the blue crab portion, and really the emphasis now is just to tell you that there's a lot of this kind of information that requires different spatial analyses, and much of it is also we can do through numerical simulations and so on. So anybody who's interested in spatial statistics, this is sort of kind of data that's waiting, and it's actually publishable and will be of use in terms of restoration. Okay, in the last minutes, I just have a couple slides. So what I wanted to do is just show you the types of big data that are available for the other species. This is the thing that we're just starting in on right now. So there's an oyster reef, and this is actually a map where we're developing habitat suitability of a particular tributary here. And you collect information on depth, salinity, bottom type, and develop a suitability index for where it would be best to restore oyster reefs. So that's sort of a simplified version. Now, when we're working with subtidal reefs, it's very difficult, obviously, to map it. You can't do it through photography generally. What we do now, NOAA, National Oceanic and Atmospheric Administration, and we actually just went out yesterday to map the Lynnhaven in Virginia Beach. But they have really state-of-the-art technology for mapping the bottom. And in subtidal areas where we can't see very well, that's absolutely essential. So we're using, it's called multi-beam sonar. It's acoustic profiling, and it can get you a resolution on the order of centimeters of what's on the bottom. So we can get a picture, and what this is an actual multi-beam image of an oyster reef. In this case, it's a restoration oyster reef. You can see where the vessel probably dragged its anchor here a little bit. But this is the kind of information that we're generating now for all our oyster reefs in the bay, in the lower bay. And what we don't know for natural reefs, we're trying to figure out, is there a pattern? So we're talking about pattern formation. We're really interested whether or not, again, this is a restored reef, but we also have the natural reefs. And we're trying to figure out, is this a random pattern, or is there some pattern formation to it? Do you have a pattern formation talk at all? No? No. Okay. I thought maybe you had, so I didn't have the information, but think of zebra stripes. That's pattern formation. And there are reasons why the zebra stripes go that way. And there are physical processes, physical and biological processes that drive that. So we're trying to understand how these patterns, first of all, are there patterns there? So that's the statistical aspect. And then the second part is try to relate that to the other physical processes that determine that, because then that will allow us to restore oyster reefs in the proper geometry that they need to persist.
Just make an observation. I would think that looked kind of like a fractal pattern to it a little bit.
We've been looking at fractals. Well, we've just started, so we're just beginning to do that. But yes, we think there are reasons, or it could be a fractal nature. We're not sure at this point. It's just beginning right now. This is being done, we also have information for seagrass beds. Patches form in certain ways, and we don't know if it's random or some sort of pattern formed. So this is a wide open field using, the vessel that we used to get this information was well over $1 million, all the technology that's on there. The multi-beam unit, just that unit itself, costs $160,000, so it's really state-of-the-art technology. They also have what's called a sub-bottom profiler that can actually penetrate the sediment, and it can tell you what's beneath the sediment. So we can see that some of the ancient oyster reefs, we've actually determined that they can be on the order of 40 to 50 feet deep below the sediment. That's how much the oyster reefs grew over time, over historical time. So anyway, there's a lot of information there. This is one of the really exciting parts. We're just getting started on that. And then finally, I had to put in a quick plug that, of course, we're doing all the spatial stuff, but we've got a major effort that I'm not going to talk much about to basically try to model the demography. So that's a population model of the oyster reefs. And so it's just a plug that if anybody's interested in continuing on that, we also have the whole time or dynamics aspect of the system. And that's my last slide. Okay, that's it. Okay. So, and I'll just say that if any of you are interested in doing any kind of a project on any part of the data, whether it's a small scale project or larger scale, I welcome that. You can either email me or email June Peng. He knows how to get in touch with me. So, okay? I hope that some of you, at least mathematicians and computer scientists, get into biology and ecology.
Okay, so this is our last talk. Thanks all of you again for participating in this class. So you can turn in the the summary last week. For this week, you can either turn it into my office or just email me the summary. Also, again, if you're interested in any of the projects we described in this class, I guess we can support such research in the future. Also this summer. Okay.